Hello everyone, my name is Moueden Amine. I am Associate Professor in Medical School of Oran in Algeria. Today I am going to talk about the involvement of infectious disease in acute lymphoblastic leukemia. We start with introduction. The acute leukemia is defined by the presence of more than 20% of blasts in the bone marrow according to definition of the World Health Organization. This can be classified according to the lineage in myeloid leukemia or lymphoid leukemia. Acute lymphoblastic leukemia includes the T acute lymphoblastic leukemia and the B acute lymphoblastic leukemia. The acute lymphoblastic leukemia are the most common cancer in children. Their prognosis is better in children than in adults. Acute lymphoblastic leukemia B represents more than 80% of all acute lymphoblastic leukemia. Acute lymphoblastic leukemia T are considered to have poor prognosis compared to acute lymphoblastic leukemia B. Neoplastic cells, on the one hand, prevent the proliferation of normal cells responsible for bone marrow fire, and on the other hand, they infiltrate various organs, mainly bone marrow, liver, spleen, ganglia, causing the tumor syndrome. Leukemia processes are still poorly understood. However, several factors have been identified which might increase the risk of development of the acute lymphoblastic leukemia. We can cite genetic predisposition, family forms like Down syndrome, blood disorders like myelodysplastic syndrome, treatment with chemotherapy, treatment with ionizing and no-ionizing radiation, exposure to chemicals such as benzene, and finally infections. The involvement of infectious disease in acute leukemia. Infections are the first causes incriminated in the development of acute leukemia. The impact of infectious stimuli on leukemogenesis has been previously implicated in multiple epidemiological studies, such as overlapping age patterns of childhood infection and the peak incidence of acute lymphoid leukemia that suggests that one or more infectious agents might promote childhood acute lymphoblastic leukemia. Subsequently, this hypothesis was explained by Graves and collaborators in 2006 by an inadequate immune response to early in life viral and bacterial infections. The presence of the genetic alteration seems to create a hidden pre-leukemic clone as an initiating event in utero. And the appearance of leukemia requires exogenous risk factor such as infections, who will give the abnormal immune response to infection. In this figure, we can see how the hidden pre-leukemic clone was created by an initiating event, and coming after the second event like infection, and therefore the leukemia appears. Example: the TML1 translocation 1221 translocation is the most common chimeric fusion gene in childhood cancer selectively associated with B-cell precursor acute lymphoblastic leukemia. The TML1 protein generates and maintains pre-leukemic clones. Infection may promote the malignant evolution of TML1 expressing pre-leukemic clones. In addition of the above, the TML1 leukemia fusion gene dysregulates the TGF-beta pathway in early B lineage progenitor cells. Infection exposure is a causal factor in B cell precursor acute lymphoblastic leukemia as a result of PAX5 inherited susceptibility. In the first was we must to define ID mechanism. Activation induced deaminase is responsible for the induction of secondary diversification of immunoglobulin genes in secondary lymphoid organs in response to antigen. Infections promote B acute lymphoblastic leukemia through activation induced deaminase independent mechanisms in the absence of IED, providing evidence for a new model of childhood B acute lymphoblastic leukemia development. Another possible mechanism of development of leukemia that the viruses might integrate into the genome of precursor B cells. Example: prenatal C adenovirus infections might generate the aberrant clones of lymphocytes that precede development of childhood acute lymphoblastic leukemia. T acute lymphoblastic leukemia develops in less than 5% of individuals infected with human T lymphotropic virus. The human T cell lymphotropic virus type 1 was the first discovered human retrovirus. This virus disturbs the immune response, which results in human T lymphotropic virus associated myelopathy, tropical spastic paraparesis. Epstein-Barr virus infection in Burkitt lymphoma. Epstein and collaborator firstly discerned Epstein-Barr virus in a cell line established from a Burkitt lymphoma biopsy by electron microscopy in 1964. This is the first human virus related to oncogenesis. At this time, no study has been able to prove that a very specific microbial agent is the main cause for acute lymphoblastic leukemia. However, other studies failed to detect the viruses in leukemic cell. Example: the human polyomaviruses in leukemia. This is now enveloped viruses. Gustafsson and collaborator in 2012 found that the prenatal infections with these viruses are not a risk factor of childhood leukemogenesis. The virus was not detected in neonatal blood. The human herpes viruses or human parvovirus B19 with the diagnosis of acute lymphoblastic leukemia. Country-wise, B19 virus is a particular complication in cases of acute lymphocytic leukemia. In conclusion, we can say further studies are needed to prove biologically the implication of infection in the development of acute leukemia in order to find preventive measures to fight against these infections. However, bacterial infections remain a very dangerous complication of acute lymphoblastic leukemia, which requires rapid diagnosis in order to start antibiotic therapy. And thank you for attention.